Okay, so we're going to start with week 7, again, chapters 9, 25, and 10. So first things first, chapter 9, this basically covered microbial nutrition in a very broad sense. And the idea here with chapter 9 is that we have the different types of trophes. And we talked about this in our in-person lecture, but again, just to remind everybody, when we talk about troph, we are talking basically about whether where your carbon comes from or where your energy comes from. So in the case of our trophs, we have, pull up my highlighter thing here. Try to zoom in a little bit. It's really small on my screen. With our trophs, we have the hetero versus auto, where are their carbon coming from? So it's either an inorganic source or organic molecules from other organisms. So that's a carbon situation. My pen is like not calibrated right, so it's writing in a different place than where I think it's supposed to write. Whereas our photo versus chemo is an energy thing. Okay, so again, you are a phototroph if you get your energy from light, and you're a chemotroph if you get your energy from chemicals. Okay. Then we have our growth factors. These are things that we brought up previously in other chapters. Basically, a growth factor is something that has to be provided to the organism that they cannot make itself. So for us, for example, like vitamins, they have to be provided in our diet for us because our body can't make the vitamins. So it has to be provided to us in our diet somehow. Then we talk about different types of organisms and different environments that they live in. Saprobes break down dead things. We call it detritus or detritus. I don't care how you pronounce it, as long as you can recognize it on the exam. They break down dead things. Saprobes are heterotrophs. Hetero, actually even more content than that. Saprobes are chemo heterotrophs because they're breaking down chemicals for energy and they're getting their carbon from organic molecules that other organisms have. It just happens that their source of carbon and energy is coming from dead things, as opposed to us. We're also chemoheterotrophs. We aren't eating decaying things, though. We are eating what are fresh, basically. Leave it at fresh, okay? And in the wild, we might be still basically otherwise living, but no longer combating this, for example. Or in the case of plants that we might eat, because we're omnivores, they're still, mostly living, whenever we're dealing with them. All right, so saprobes specifically digest dead things, but they're still chemoheterotrophs. They're a type of chemoheterotroph. All right, halophiles, these are organisms that flourish in salty habitats. Halo is referring to halogens. So in the chemical periodic table of elements, halogens are the ones that are on the far right, and that includes chlorine, for example, so sodium chloride. These are organisms that can live in the presence of salt. Okay. Thermophiles, these are organisms that grow well at high temperatures. We're talking even above boiling in some cases. We call them hyperthermophiles, but they are still thermophiles. All right, the next part talked about diffusion. Diffusion is movement of molecules from an area of high concentration to low, or spreading out the molecules, right? And that can happen very gradually. Like we said, for example, in class, if I spray perfume, then the molecules will eventually diffuse out throughout the class and the whole room. And that it can be very gradual. So once I spray that perfume, maybe after an hour, it might have spread out so much that you no longer even smell it in the area. Or it might have gotten so spread out, you only vaguely smell it very, very far away from the original spray point, and been vaguely everywhere else as well because it's spread out so much. That's diffusion. And diffusion can be any molecule. But when we talk about osmosis, osmosis is a type of diffusion. its diffusion of water. And this is because the water molecules can move across our lipid bilayer because our lipid bilayer is selectively permeable. It allows some things to cross over it and not others. All right, so some small molecules might be able to cross the membrane, but generally speaking, when we think of the cell membrane, generally speaking, water can cross in and out of the cell, but other molecules like sodium chloride, they can't. Oh my gosh. I moved this so far away. It's too close. All right, there we go. Sodium or chlorine can't go in and out. So only water can move. So if you have a cell and it's got some stuff on the inside and the outside has more of that stuff, the stuff, doesn't matter what the stuff is, cannot move in or out to equalize things. The stuff can't move. Water can. So water would come out in this situation because this solution outside has higher amounts of the stuff. In this case, the stuff is what we call a solute. What the stuff is dissolved in this case, it's usually water in biological systems. It's called the solvent. In order to balance basically the concentrations out on both sides. So if the stuff can't move in and out, like when we pack a cell in salt, the stuff, the non-water stuff, more stuff outside of the cell than inside, which causes the water to seep out. This is a situation that we would call hypertonic on the outside. If we have the outside is higher, that means we are hypertonic. Hypotonic would be, for example, if I had a cell, we'll draw him purple so we're away from the stuff. We had a cell that has stuff in it, and it's in a cup of pure water. Now, which way is the water going to flow? The stuff can't move. These things inside the cell can't move across the membrane. Chemistry and physics said, okay? So if that can't equal things out, then what happens is the water moves in that situation. This is a situation that we call hypotonic because the stuff is at a lower concentration on the outside. Where is it? Here it is. All right. In order to move the stuff, the non-water stuff, sodium chloride, sugars, proteins, whatever you want to move in and out of the cell. If you want to move things in and out of the cell, it can't just cross the membrane on its own. Impossible. Okay. Only the water can do that. We already said that was the rule. Only the water can move. So if you want to move other molecules, you're going to need to use facilitated diffusion or active transport. Facilitated diffusion is something where we build a channel that helps the things move across the membrane. It's usually activated by the presence of the things. And it usually still goes from area of higher to lower, like normal, moving with the gradient, normal movement of the molecules, if we're doing facilitated diffusion. It's once we get into active transport that we can move against the gradient and move things in and out of the cell against the gradient or at a faster pace than what we would see if we were moving things just naturally. And I went over a lot more detail on this in our class review if you haven't watched the videos for that, or if you didn't attend one of those classes. Trying to clean up my screen so we have more space to write stuff. Nope. All right, what's next? Temperature tolerance. Some organisms grow in low temperatures and some of them grow in high temperatures. Some of them can survive and tolerate low temperatures or survive and tolerate high temperatures. So if you are tolerating versus whether you're thriving at the temperature can also define you. Psychrophiles is a term where they actually grow in the cold. All right. Mesophiles are organisms that grow at what we consider neutral room or body temperatures, the middle range temperatures. Facilitated diffusion doesn't really need energy in the traditional sense. So that's a good question, Gracie. So Gracie asked the question, facilitated and active transport, do they both need energy? But active transport goes against the gradient, while facilitated diffusion goes with the gradient. Facilitated diffusion may need energy depending on the system. So that one's a little bit trickier. So we can have, this is your membrane, and this is your channel. Your channel. Doesn't look like much of a channel right now because it's closed. Your thing comes up to it and binds to it, and then that causes that channel maybe to open. I don't know, I'm drawing it weird like this, but that's okay. We're drawing it like this, okay? And now the thing can move through. That can cause the change to happen on its own without adding energy to it. Or sometimes we might need to use ATP as an energy source to allow that to happen. But facilitated diffusion does always go with the gradient. So the movement of the molecule moves, the molecule itself moves because of the gradient, but the opening of the channel may or may not need energy. In active transport, we always need energy. Basically, and we're going either against the gradient. Oh my gosh. We're either going against the gradient in active transport, or we're speeding up the gradient. So let's say you're a cell, and you want to eat this food out here. Well, if you open up your little channel to let the food in, here's your channel. Oh my God, I've erased the whole cell. Don't you love it? I'm trying. So you've opened up your channel to let the food in. If you let diffusion just want to do the work and let the molecules of food come in on their own by diffusion, that might be too slow for you. So if you want to speed up the thing, then you're going to need energy to speed up, but it might still be, the gradient. Also, let's say you're a cell and you want food, but you've got a lot of food in you, but you still want more in you. Now we're going against the gradient, and that would need energy too. So it just basically dictates that. So active transport always needs energy, may or may not go against the gradient. If we're relying on the gradient, if we say that there's still, the gradient, it just means that we want to go faster than what natural diffusion would allow to happen. So we need energy to do that. Facilitated diffusion relies on the gradient always, but then it's a matter of, does the channel need energy to open or close? That may or may not be the case. Does that answer your question, Gracie? Hope so. Awesome. Perfect. Okay. That was a good question. Okay. All right, so back to the temperature tolerance. We got our psychrophiles. They like to grow in the cold. The mesophiles that grow at like the temperatures we exist at. So all of our pathogens typically are mesophiles. They typically grow at the same temperature we do, the middle temperatures. And then we have the thermophiles that prefer it to be hot, including they can actually keep multiplying even at boiling temperatures. It's like the guys that live in the deep sea vents, like those guys. Then we have how we classify organisms based on how they deal with oxygen. And that led into our discussion of those toxic oxygen products. We call them reactive oxygen species. We actually had a question about it from one of the students at my night class when we were giving this lecture. She joined in on that class and was asking for me to go over this. So we'll briefly discuss these guys. Whenever you are dealing with aerobic respiration, in aerobic respiration, which is chapter 10, you're right. The final electron acceptor in aerobic respiration is oxygen. And what do we do with oxygen in aerobic respiration when we finally get it? The whole point of it is to take some electrons with a proton and turn it into an H. We get two of those regular H's with electrons in them, so that they can share electrons with half of the O2, which is oxygen, which makes water. Sometimes when you're splitting that O2 up to get half of it out, we might do that unevenly. We might have extra electrons get added on and all sorts of stuff. So we can create reactive oxygen species like O2 with an extra electron, or H2O2, which is hydrogen peroxide. H2O2 is hydrogen peroxide. O2 minus this is O2, but we got an extra electron with us. This is superoxide. And there's other ones too, like ozone and stuff like that. And all of these are just versions of dealing with oxygen in general, often because of aerobic respiration, but it can also just be from oxygen in the environment. We can have generation of these things too. And these things cause oxidative stress. They can cause other molecules to lose electrons. Remember, if you're oxidized is loss. Oxidation is loss. Reduction is gain. So if you're losing an electron, you're oxidized, right? Oil rig, right? All right. So this can cause other molecules to lose electrons, or it can lead to gaining of electrons where you're not supposed to, or losing of electrons. And this is something we'll call oxidative stress. And so dealing with oxygen means you have to be able to deal with the oxidative stress that comes with oxygen. And so this happens from aerobic respiration as well as just living in oxygen. And so we can actually define our organisms based on their ability to just deal with oxygen. So in this case, we have an enzyme that breaks down superoxide into H2O2. The enzyme that does that, do a different color. The enzyme that converts superoxide into H2O2. You might be like, well, isn't H2O2 also bad? It is, you're not wrong, but we'll get there, okay? The enzyme that breaks superoxide into H2O2, we call that superoxide. Sorry, I wrote off the thing. Dismutase. And then another thing can break hydrogen peroxide, which is H2O2, into H2O and O2. I know the equation isn't balanced. I think we'll be okay. Don't tell Dr. Tim that I said that. He's probably like literally sitting inside of my door right now. Don't tell him I said that. Okay, catalase. Catalase catalyzes that reaction. So turning hydrogen peroxide into water and oxygen, which is why whenever you put hydrogen peroxide on a wound, it bubbles because the gas you're making is oxygen. That's safer for us to deal with than superoxide or hydrogen peroxide. Deal with oxygen. So those are the enzymes that allow us to deal with those reactive oxygen species. There are other things that help us deal with other aspects of other versions of those guys, but these are the main two that you need to know about. And it will come up again later in other units. All right. So if you deal with oxygen and you use oxygen in aerobic respiration, you're one of these guys, it's aerobic respiration, you are an aerobe. If you are a facultative anaerobe, you will do aerobic if you have O2. If you don't have O2, then you will ferment. Overall, as a species, as an organism, we are aerobic. We have some of our cells that can be facultative, but generally as a whole species, we're aerobic, right? So some of your muscle cells can undergo fermentation and make lactic acid, which is where you get lactic acid buildup when your muscles don't get enough oxygen to them. If you're working more than what your mitochondria can keep up with, then we'll get ATP out of glycolysis and just ferment. All right, that's what tells your body to make more muscle cells and more mitochondria. Microaerophiles need less oxygen. Aerotolerant anaerobes do not use oxygen, but they can survive from it. They still can deal with it with superoxide dismutase or catalase. They can deal with oxygen, but they don't use it in aerobic respiration. They go through anaerobic respiration. Then we have obligate anaerobes. Obligate anaerobes do not use O2 either, but obligate anaerobes also can't deal with it, with any of those reactive oxygen species. So they die in the presence of oxygen. Oxygen and its reactive species kill them because they can't manage it. It's too much for them. Some organisms that are considered obligate anaerobes include all Clostridium species. Clostridium botulinum, Clostridium tetani, Clostridium perfringens that causes gas gangrene, as well as Clostridium difficile, C. diff. Those organisms die in the presence of oxygen. However, we also learned that Clostridium species can form endospores. And the endospores are hardy enough that allows the organism to survive being in the presence of oxygen until they get into your deep tissues, where they can wake back up into the vegetative version and start going back into being anaerobes again, vegetative anaerobes. Makes sense? I hope so. Okay. That leads us into our symbiosis discussion. Clear all this up. Symbiosis is whenever two organisms' livelihoods are intertwined somehow. This means that the relationship is required for at least one of the organisms. So if you have two organisms, at least one of those organisms must have that relationship happen for them to survive. It's not optional. It's not like you and your dog, like we said in class. It's not like you and your dog. You might give food to your dog. So you guys have a relationship, but it's not symbiotic. Okay. In a symbiotic relationship, your dog would die if you didn't feed it. But your dog isn't going to die if you don't feed it, because it can survive on the street and get food there, or it can get, food from, eating the bugs in the ground. Let me tell you guys, Meatball loves eating bugs in my backyard. It was ridiculous. Like, he got fat from it for a while. wasn't me feeding him. I gave him very little food, and he would eat so many bugs in the backyard. He got chonky, okay? He loves eating the bugs. He used to live on the street, and that's how he lived. He's like part Chihuahua, and he is like just a little bit bigger than a Chihuahua. So it wasn't like it doesn't take a lot of bugs. Just saying. So that's not a symbiotic relationship, but you get bit by a tick. The tick has to feed off of the host in order for it to live. That is symbiotic. There's three different types of symbiotic relationships that you can have. Commensalism, mutualism, and parasitism. Sorry, my brain. Okay, so in mutualism, both organisms benefit. I don't know why I skipped commensalism. In mutualism, both benefit. In commensalism, one benefits. The other is unaffected. No harm, no benefit. The one that is benefiting needs to benefit from what it is benefiting from. The other organism is unaffected. All right, parasitism. One organism benefits. We call this the parasite. And the host is harmed. Those are the three types. Both benefit. One benefits, nobody is affected. One benefits, the other isn't affected. Or one benefits and the other harms. Not both harmed. Nobody would that's not a symbiotic relationship. Nobody would exist because you're both being harmed. So it's these, okay? The term obligate parasite. Obligate means obligate. So if you are obligated, it means that you have to do something. So you are obligated to go to the bathroom, a couple times a day. You have to do it. You are obligated to breathe, right? You are obligated to eat and things like that. You have to do those things. Obligate parasites are obligated to be parasites. That means they are parasites regardless of the situation they're in. Fungi, for example, some molds. Molds might be saprobes most of the time. They get into your sinuses, they might become a parasite. That's not an obligate parasite. That's an opportunistic parasite. Obligate parasites must be parasites. They just are, like viruses. Viruses always have to be in your cell and using up your cell's resources. They're causing you harm, and they're benefiting from causing that harm, and they have to live like that. They are obligate parasites. Okay? They can't survive outside of the host, separate from the host. All right. Then finally, we had biofilms, where we talked about quorum sensing. In quorum sensing, the idea is that the organisms that are living in the community together can basically sense what is going on in that community and then are able to adapt accordingly. So what does that really mean? That means that there are chemical signals being given off pretty much at all times just from living. And what I mean is, if I'm living here in my office, actually the better example that I gave in my class was that we live in a building together. So living in a biofilm is like if we were living in a building together. So we're all living in a building, and here's the shape of our building. It's a nice gooey building. Go figure that it would be. And then these organisms are living at the bottom. Now, this building, we're living at the bottom of the building. And these organisms are living at the top. And there can be many, many layers in this building, many floors to the building. Wow, if these guys down here in the bottom start cooking, the people in the top might smell that and say, smells like food. Might go over and have a meal with them. Stuff like that. Or they start smelling smoke and a lot of it. They might be thinking, oh no, we need to get out of the building because there's a fire somewhere in the building. Those are chemical signals, and the organisms in the environment can respond to them. So there's those kinds of chemical signals that are just being given off by experiences as waste for example, or the presence of chemicals from food or something like that, or toxins and stuff like that, that the organisms can, number one, respond to that in their building, in their biofilm. Or number two, if this guy up here smells the smoke, he can send signals to his friends as chemicals that it produces saying there was danger nearby. So danger signals. All of this is what we would call quorum sensing. Responding to your environment and then sending chemical signals that alert your neighbors to what is going on in your environment. We can send danger signals if we smelt that smoke. Or this guy, no, I clicked on this. Or this guy might smell food and then send signals to his neighbors saying, hey, come get food. That's quorum sensing. And again, it's just by producing chemicals as signals. We produce sounds that you can interpret to mean certain things, but these cells aren't that smart. So they make a chemical that serves as a signal to the next cell nearby it, and that cell reacts by binding to that signal molecule, causing proteins to change shape. And when the protein changes shape, that lets the cell know it needs to move towards a thing. If protein changes a different shape, it lets the cell know it needs to move away from a thing, for example. So that's how quorum sensing works for those cells. That's in biofilms. Quorum is a group. And so, for example, I'm on Faculty Senate, which is where I was right before this meeting. And so we can't conduct business unless we have quorum, which is the amount of people that are supposed to be there to do business. Quorum is just a group. This is like sensing your group, sensing your population. Okay. All righty. What else do I want to say? The growth curve. This is the growth curve. Oh no, why are you doing that? The lag phase, you're lagging. You're not growing. This is a period of time where we are just adapting to our new environment. We're realizing we have food or water or shelter. And it takes time for the cell to decide, I'm going to go ahead and replicate myself because things are good. So that is a period of time that we call the lag period. It's a period of time where when I take my bacteria on my loop and put it into fresh media and swirl it in there, the time between when I swirled it in there and they start to actually divide and make new cells, that's the lag period. So it's when we're adapting to the environment and when we are making copies of the DNA. You can't make a new cell without copying the DNA first and then splitting, right? You copy your DNA and your other cell parts, like new membrane, new ribosomes, whatever else you need to make for a new cell. New peptidoglycan for your wall, right? So that's the lag phase. The next phase is the log or exponential phase. This is a phase of division where one cell becomes two cells and each of those splits and becomes two cells. This should look a lot like PCR. Doesn't it look like PCR to you? Because it should. It's the same concept. This is how our cells divide too. One cell becomes two, divides into two. One cell doesn't divide into three cells. One cell divides into two cells. Each of those becomes two. Each of those becomes two, and so on and so forth. That type of multiplication of cells, we call it exponential growth, because what's happening is division by 2 to the n. It's exponential. Okay, it's an exponent in case you forgot from them. Okay. All right, the next phase, this is when we're dividing. In this situation, the log phase, exponential, we have all the resources we're needing, and we're dividing at our max rate. Making new cells. The next phase is called the stationary phase. And it is what it sounds like. It's not that nothing is happening in this phase, but what's happening is that the number of cells that are new cells is roughly equaling the number of cells that are dying. Roughly. And so we're not increasing our population anymore. We're at a stationary level. And maybe we're not dividing as quickly because there aren't as many nutrients available. So this is where we're leveling out. And then obviously that last part is the death phase, where now the cells are dying off faster than they're reproducing by a lot. This is when we run out of nutrients or there are too many toxic waste products building up and stuff like that. All right, any questions about chapter 9? All right, moving on to chapter 25. In chapter 25, we go over the carbon cycle as well as the nitrogen cycle. I'm not going to talk about the carbon cycle because I feel like, well, we'll briefly just say what the carbon cycle is, okay? We have carbon dioxide in the atmosphere. Carbon dioxide in the atmosphere gets fixed into place from plants. Here's a tree or broccoli. It doesn't matter, they're both plants. And plants convert that carbon dioxide into, my pen is weird, I'm sorry, I apologize. Plants convert CO2 into organic compounds like glucose. I thought I had some stuff. Okay. Remember, carbon dioxide is inorganic. I don't know who has been telling you guys, right? I don't know who has been telling you guys that carbon makes something organic, but they's lying to you. I know Dr. Tim didn't tell you that, and I know Dr. Singh didn't tell you that. You're only organic if you have hydrogens bound to your carbon. You have to have carbon, but you have to have hydrogens bound to your carbon to be organic. So again, carbon dioxide is inorganic carbon. Anytime you have hydrogens bound to your carbon, organic, okay? So plants take inorganic carbon dioxide from the atmosphere, turn it into organic molecules. And then other organisms eat the plants, like, I don't know, dogs. Here, this is your dog. This is my doggy. This is Meatball. And he'll eat the plants. He'll eat anything, dude. He'll eat the plants. And then he'll exhale CO2 back into the atmosphere. And then we start it all over again. Carbon cycle is easy. Anybody want more explanation than that? Obviously, combustion and things like that also make carbon dioxide and feed into that cycle as well. Okay. Nitrogen cycle, which is the thing that a lot of people hate. We have nitrogen in the atmosphere. And then some bacteria can turn that nitrogen in the atmosphere into ammonia. This is the ammonium ion, all right? That's enough for me, okay? We call this process of turning atmospheric nitrogen into ammonia ammonification. We then have some bacteria that turn ammonia into nitrite. And then can turn nitrite into nitrate. This process is known as nitrification. Bacteria that live in a plant roots can do this. And then when that happens, we call it nitrogen fixation. I'm sorry, that was loud. We call it nitrogen fixation. Bacteria that do that in a plant's roots, because plants can use nitrate. Plants can't convert atmospheric nitrogen into nitrate, and they need nitrate. And then they can convert nitrate into things like your DNA, RNA, and protein nitrogen sources. And then we can eat the plants and get our nitrogen from the plants, right? But the plants can't get the nitrogen unless the bacteria turns it into nitrate. Okay? And then we have bacteria that will turn nitrate back into N2 atmospheric nitrogen. And that process is called denitrification. I know I wrote off the screen, but it is on the thing here. Here it is. That's all I need you to know about it. I don't need you to balance the equations or know what their intermediates are or anything fancy like that. Have a sense of where each of these terms plays a role in the nitrogen cycle. Okay. Okay, plastic creates islands or reefs where microbes can kind of grow and attach onto them and also block the sun from all of the organisms beneath those islands that usually need the sun. So things like plankton and algae and stuff like that need sunlight in order to be able to live. And then other organisms need to eat those things in order for them to live. And so plastic islands, reefs, whatever you want to call them, all interrupt that process. So obviously that's bad. When we think about plastic polluting our environment, what does pollution actually mean? It means literally creating a physical barrier from interacting with our environments, whether we're stacking it up in landfills and then it's blocking all of that ground from getting sunlight and oxygen and things like that, as well as doing the same thing in our oceans. So it can inhibit those environments by blocking them from the resources they usually interact with. All right, and then moving on to water filtration. In water filtration, we might filter water for drinking with normal, typical water filtration. We get water from a river or an aquifer or a lake or whatever, and then we filter it for drinking and consuming versus wastewater treatment. This is treating our wastewater. What is wastewater? Pee and poo water, sewer water. Okay, so we treat that. We don't just pee and poop and then you flush it into the river. That isn't how it actually works, okay? That is how it works in some countries that don't have proper water treatment and sewage management systems, right? And that's why they have the diseases that we don't have. So things like helminth infection and other fecal oral route types of infections like cholera. But we treat our wastewater to release it back into the environment so that we're not re-exposing ourselves to the organisms that are associated with the diseases from those environments or from that fecal oral waste. So we usually do that through multiple steps, including removing the solids, filtering it through several steps of filtering, and then typically treating it with UV radiation or chlorine. We can bubble chlorine gas through there or add chlorine, basically molecules back in to deal with that. And if you watch my videos and you already know how I feel about Bethany water, and if you want to hear about that, please go back and watch one of those videos. I'm not going to go back over it again. But also always remember, once we treat the water at the water treatment plant for you to drink, which is this kind of first layer of stuff, right? Once we treat that water, it travels through miles of pipe that has been in the ground for decades. You think they clean in those pipes? I don't know what to tell you. Okay? Yes, I still drink tap water and I still drink from water fountains and stuff like that. I'm just saying that water is not clean, not sterile when it comes out of your tap. It is not. Either way, whether we are treating water for drinking or treating water to return it back into the environment through wastewater treatment, we will look for coliforms. Coliforms are indicator bacteria. They're always bacteria. They're indicator bacteria. For example, we know you have normal E. coli in your colon. There's other organisms that live in there too, like Enterobacter species or Klebsiella or all sorts of stuff that can live just normally in your colon. If we see any of those normal colon bacteria in our filtered water, that indicates to us if the normal bacteria got there, the pathogens could get there too. So coliforms is looking for normal bacteria. If the normal bacteria can get there, so can the pathogens. And we don't even need to test for the pathogens. If the normal bacteria got there, we're going to assume the pathogens could get there. That's more concerning than testing directly for the pathogens. Okay. So we use coliforms as an indicator. Coliforms are bacteria. Let's say you test the water and you have no coliforms in it, no bacteria grow at all. How do you know you filtered the viruses out? Viruses don't grow on an agar plate. They grow in cells. We don't test for viral presence in our water the same way. We will test for certain viruses through PCR and things like that and see if those viruses specifically are in the water as needed when necessary. We don't do it necessarily all the time. Okay. So we have to be mindful of our filtering practices, be aware of, is there risk for viruses getting into our water supply? With COVID, there was, and that's what that activity for chapter 25 was referring to, was COVID potentially getting into our water supply. And so now we have to treat our water filtration process to eliminate viruses that got into our environment from our wastewater and getting back into our consumption water and being mindful about that. All right. Obviously, we use microbes in food preparation, fermentation, and preparing all sorts of nice goodies and things like that, as well as the effects of microbes on our environment, not just causing disease. This chapter also talks about One Health, which is basically talking about the health of the environment and how it affects all of the living systems altogether, not just thinking about human populations, but the health of our ecosystems altogether. As we are moving more and more and encroaching into wild territories and building up urbanization, basically building up into new areas, we are encountering microbes in those areas that can serve as pathogens. So we're seeing new pathogens that way. Also, as we're living closer and closer together, we're also being exposed to microbes that might not be transmitted if we were living further apart. And so we also see reemergence of infections like tuberculosis. Tuberculosis was obviously a big thing back in the day. If you've heard of consumption, tuberculosis is consumption. That's tuberculosis. And that used to be a big bad thing back in, the early 1900s and before. And then we started eradicating it once we started having antibiotics. Now it's coming back again because we have people living in close proximity who also tend to have HIV or other diseases that can debilitate the immune system. And the tuberculosis is becoming antibiotic resistant as well. And so we're seeing reemergence of it in our civilization now. That's just one example of a reemerging disease. There are several. All right, any questions about chapter 25? All right, next chapter, Brian. Obviously, this is the big chapter. It's chapter 10. I'm not going to go into a lot of detail with this stuff. Again, you will watch the lecture video from my other class, and you will see me talk about enzymes and all of that stuff. So briefly, to touch on this stuff, metabolism is all of the chemical reactions in a cell. If you're breaking bonds, you'll release energy. That's catabolism. Breaking molecules to release energy. Anabolism is using energy to build things, right? And amphibolism is whenever you link those two together. You break bonds, you collect the energy, and you use that energy to build other things. That's amphibolism. Okay, amphibolism, both sides of the bolisms. Okay. All right. Enzymes. Enzymes are protein catalysts that help lower activation energy. Activation energy is the amount of energy that you need to make a reaction happen. If you're trying to convert carbon dioxide into glucose, the amount of energy that requires is called the activation energy. And enzymes help decrease that by helping the process happen. They're not usually used up. You can reuse them over and over. We can reset them and use them over and over and over again. The molecule that an enzyme acts on is called a substrate. So if this is your enzyme, then it acts on your substrate, another pizza slice. The substrate binds at the part of the enzyme called the active site. You can have simple enzymes that are just protein, or you can have conjugated enzymes. Again, enzymes is what we're talking about here, okay? Trying. Conjugated enzymes consist of a protein portion, which we call the apo enzyme, and a non-protein portion, which we call a cofactor. That cofactor, can be organic. If it's, I'll put an O for organic, then we would call it a coenzyme. If you're inorganic, you're just a cofactor. You don't get a special name. So just think of a coenzyme as being a more specific cofactor. An organic cofactor. Okay. The whole functional apoenzyme plus the cofactor together, combined, create the holo enzyme. The whole enzyme is the holo enzyme. Okay. So that's the idea of all of this stuff kind of up here. Talk about the different enzyme classes. The different enzyme classes, basically based on what their overall general functions are. Different enzyme classes were oxidoreductases, which are involved in transfer of electrons. Then we had transferases, which transfer groups from one molecule to another. Functional groups of some kind. Then we have hydrolases, which basically break water in order to break bonds. We have lyases, which involve double bonds, which lead to that. Isomerases, which change the shapes of other molecules. And then ligases. Ligases like DNA ligase. They join things together, creating bonds. And they actually, usually when they join things and then create H2O as a product. We'll say as a byproduct. Whereas in hydrolases, we broke things. We break things and also use water. We also break water while we're breaking other things. So they're like opposites. Hydrolases and ligases. They're kind of like opposites. Okay. Alrighty. All right, denaturation. We covered this pretty well, I feel like. The idea of denaturation, it's based on the fact that proteins and their shape are held together by hydrogen bonds and ionic bonds and disulfide bonds. And heat and pH and chemicals can break any of those bonds, therefore disrupting the shape of the protein. We know we can denature proteins, as I just told you that. You can do the same thing for DNA, which is also held together by hydrogen bonds. So both proteins and DNA structure can be denatured. We know that because we learned about PCR and denaturing DNA with heat in PCR. It's the same concept. Hydrogen bonds also hold a protein together. You can break them with heat, right? Right. That's denaturation. Inhibiting your enzyme. Again, if this is your enzyme, then you normally bind to this. Normal activity, this is your substrate. And this was your active site. Right? Then, in the case of competitive inhibition, it's competitive because we're competing for the active site. In this case, your inhibitor, okay, whatever, why it's doing that. Binds at the active site. So now your substrate can't bind there. We competed for the active site. In non-competitive inhibition, what happens is you have your enzyme, your inhibitor binds somewhere else. I'm getting some phone calls today. And that causes the enzyme to change its shape. Maybe it closes its little mouth. And now the substrate can't bind there anymore. We blocked somewhere else. We bound it somewhere else. That place where we bound at it, we call it the regulatory site, or also known as the allosteric site. You can also control enzyme expression at the genetic level, like we talked about with operons. You can block the promoter region. You can block it at an operator so that the RNA polymerase can't go. And we can also have the turning on and turning off of genes at the DNA level. So this is all happening, competitive and non-competitive inhibition is happening once you make the enzyme. This is acting on the enzyme itself. But when we talk about activating the genes associated with making the enzyme, that happened at the DNA level. So those are all ways you control proteins. But are you controlling the expression of the protein by controlling the DNA that codes for it? Or are you controlling the protein itself? And so that's what this inhibition stuff talks about, the protein itself. Alrighty, next thing. Redox reactions, we have exchange of electrons. If something loses an electron, something else gained an electron. So our way of remembering that was Leo says GER or oil rig, whatever you like. Leo says GER. Lose an electron and you are oxidized. Gain an electron and you are reduced. Oil rig, whichever one you like best. I learned Leo says GER, but you do you. Oxidation is loss. Reduction is gain. Oil. And so I use this to remember which one is which. And so anytime we're exchanging electrons, we can track where the electrons go, and we can talk about which one is gaining electrons and which one is losing electrons based on oxidation and reduction. For example, if you have sodium chloride, we know that the sodium donates its electron to the chlorine, right? And then it becomes Na is positive because it lost an electron, and Cl is negative because it gained an electron. Sodium lost an electron, so therefore lose electron, oxidized. Chlorine gained an electron, you are reduced. And this is ionization. You don't have to go through aerobic respiration to see redox reactions. Redox reactions happen all the time. So it's what basically governs our ions. So that is redox. And then that leads us into talking a little bit about ATP. ATP is the energy molecule of the cell. How does it store energy? And when I ask students that, a very common answer is to say, oh, well, we broke down glucose and then turned it into ATP. Yes, but what does ATP do after that that gives energy to the cell? What is it doing to give energy to the cell? How is it giving energy to the cell? Well, ATP is a sugar and a phosphate. Does that look familiar? Should. What does DNA look like? Sugar, phosphate, sugar, phosphate, sugar, phosphate. And each of those sugars is connected to a base. Well, as it turns out, so is ATP. It's an A connected to a sugar, which is ribose. In DNA, it's deoxyribose, but in RNA, it is ribose. So this is a ribonucleotide, and it has a phosphate and another, again, phosphate is technically PO4, okay? And another phosphate, and another phosphate. 3 phosphates because it's a TP. Okay, and these phosphates are negatively charged, and opposites attract, not samesies attract, okay? And these are samesies, and so these are repelling each other. But they're also covalently bonded, meaning they share electrons. When I told you guys that you weren't done with chemistry, okay, so anyways, talk about it every day, or do we talk about it every day? So anyways, these phosphates don't want to be linked together. They hate that they're linked together. They're repelling each other, but we force them to link together. So therefore, these bonds have a ton of potential energy. It is just like if I have a heavy, I don't have anything heavy. I don't want to drop this, so let's not pretend, okay? If I have a marker that I hold like this, and I hold it like this is storing potential energy. Watch me release it. Just like that. The pen wants to fall. And when I let go of it, it does fall and it releases that energy, and it's no longer storing that energy anymore. It's the same between our phosphate groups. They want to be apart from each other. We're forcing them to stay, just like I'm forcing the pen to stay right here. As soon as we break the bond, it releases the energy, and the cell can use that energy. It often uses that energy by moving the phosphate to something else. And now we're moving a negatively charged thing to something else. And that can cause the something else to change its shape in response to that negative thing being added to it. Often that's what drives our reactions. Okay? All right. So that's how ATP serves as an energy source, because it's carrying around that negative phosphate. It's carrying around 3 of them. And they will want to be near each other. And so moving those phosphates around often drives our reactions, because the phosphates have a lot of potential energy stored in the bonds between them and ATP. All right. So that leads us into aerobic respiration. I'm trying. Hold on. Give me a second. What is this? Why is this? This is not letting me delete this. Give me my cursor. Give me a second. I know you're not seeing what I'm seeing. You're going to, so don't freak out yet, okay? Here you go. All right, so now we will talk about aerobic respiration and all of that. So again, just to remind you guys, we have aerobic respiration, anaerobic respiration, and fermentation. In aerobic respiration, you go through glycolysis, Krebs, and the electron transport chain. In the electron transport chain, you will convert, oxygen into water as your final electron acceptor. So oxygen is your final electron acceptor. It accepts the electrons with hydrogen. And then because oxygen is becoming H2O, it's being reduced because we're gaining electrons reduced. So it is oxidizing. I know, don't worry, I'm not going to test you on that. Okay. So anyways, but yes, it's accepting the electrons. All right. Same thing with anaerobic, glycolysis, Krebs, and the electron transport chain. All right. In the electron transport chain in anaerobic and being without aero being oxygen. This means that our electron acceptor is anything but O2. Just something else, and you don't need to know what right now. Okay, it's not oxygen. In fermentation, we get stuck in glycolysis. We don't have Krebs or the electron transport. Are we ready? I hope we are. It starts off with glycolysis. In glycolysis, the whole goal is to make pyruvate. Promise. Okay. If you're going through aerobic respiration, which is what I'm drawing here, separate units, then glycolysis goal is to make pyruvate. How do we do that? We take a 6-carbon glucose, which is C6H12O6, and take it through some steps. I mean, it's a lot, right? But you don't need to know them. We'll take it through some steps to make 2 3-carbon pyruvates. That's the goal. While we're doing all of that in all of our steps, we will make a little bit of ATP, 2 net. You'll make 4, but you'll use 2 of them. So you make 2 net, okay? And you'll also make some NADH, but your goal really is pyruvate. These are just happy icing on the cakes, okay? NADH represents the H is the electron. That's what NADH really is. It's an electron carrier, NAD is. And NAD carries the electron in the form now of an H with its electron. It's really an electron. It's an electron carrier. All right. So the whole goal was to make pyruvate. We just happened to also make a little ATP and some NADH. Next step is the intermediate step. In the intermediate step, the goal is to make acetyl coenzyme A. Coenzyme, do you remember what it is? A coenzyme is a non-protein organic cofactor, right? So it's acetyl coenzyme A. So we're going to take the 3-carbon pyruvate, each one. So you get to do this twice because we have 2. So the 3-carbon pyruvate, and then turn it into 2-carbon acetyl CoA. It's going to give off a little bit of CO2, obviously, because we're going to from 3 to 2 carbons. We lost a carbon. It comes off as CO2 that you exhale. Okay. We'll also make another NH, which is just an electron. All right. We're tracking? I hope we are, because we're moving. If you want more explanation in these steps, please watch one of my in-class videos. Okay. Next, and like I said, you don't need to know the intermediate steps or balance your equations or anything. I'm not asking you to know that. All right. Finally, I say finally, we're prepping. This is all just prep. The Krebs cycle, also known as the citric acid cycle. In the Krebs cycle, we are taking our 2-carbon acetyl CoA. The fact that it's 2 carbons here is the most important thing. We're adding it with something that is 4 carbons. It happens to be oxaloacetate. I don't care if you know that or not. Oxaloacetate is 4-carbon thing plus this 2-carbon thing. What does that make? It makes a 6-carbon thing. Again, I don't care if you know this, but it happens to be that the 6-carbon thing is citric acid or citrate, however you want to call it. Okay. Happy. I'm not going to test you on it. You're welcome. Then we turn that 6-carbon thing through a couple of steps. Also, you are welcome. And turn it back into a 4-carbon thing. Well, if you're going from 6 carbons to 4 carbons, what happens to the other carbons? You already know. You spicy little thing, you already know what it is. We're going to give off some CO2 a couple times. In the process of all of this, we are also making a little ATP. Okay, make a little ATP in this too. But that's not the goal. The goal of the Krebs cycle is to make electrons, to collect electrons in the form of NADH and FADH2. So while we're knocking off our carbon dioxides, which is just waste, and some ATP, which is just a happy bonus, we're also making NADHs a couple times. And some FADH2. These, to remind you again, are really just electrons. Because you know what's coming next. It's the big daddy of them all. And clear some of this up. Make space. We need space for the big daddy. All right. Now, the Krebs cycle, the whole point of this was to collect electrons. We broke down glucose to make pyruvate so we could turn it into acetyl-CoA so we could steal off electrons. The whole reason we need electrons was the electron transport chain. Period T. Okay, so here we go. Here is, try to make sure what time it is. Won't have a ton of time. Here's your membrane. This is your membrane. This is either in bacteria, this is your inner membrane, or cytoplasmic. Or it is your mitochondria cristae. Which is the inner membrane of your mitochondria in eukaryotes. Okay. On that membrane, we have some, I'm going to make them yellow because I have a highlighter here, okay? We have some proteins. These proteins are called cytochromes. Really wish I had another color here. And at the end of all this, you have this happy guy. And he is ATP synthase. Again, this is all on the membrane, okay? I'll remind you, it's all on that membrane. So what happens is we take those electrons from, go away, NADH or FADH2. We take the electrons out, and the electrons move through these proteins. Well, if you're moving these negatively charged electrons through a protein, if proteins are made, their shape is dictated by hydrogen attractions, hydrogen bond attraction, as well as ionic bonds and stuff like that. Moving a negatively charged guy through them changes their shape. And then that leads to us basically taking the leftover. If you removed the actual electron, what are you left with? The H's. So as the negative thing moves across the protein, it allows us to move the H's out. And we build up a ton of the H's out here. This is called the proton motive force. They are building up out here, creating a gradient. And we already learned with diffusion, but they want to equal out on both sides. The only way back in is through ATP synthase. And if a negative charge caused a protein to change shape, a positive charge will too. ATP synthase changes shape because the positive thing is moving through it. When it changes shape, it allows it to turn ADP into ATP by adding on phosphate. Now we're building up excess H pluses. And we got to move those electrons through too. We got to get them out of there so we can keep moving them through. So if we take an electron plus some H pluses, we just turn them into regular H's. Then we take a half of an oxygen, which is just an O. We combine this all together. Regular H, a regular H, carrying their electrons with them, can now share those electrons with one of those O's, and you have H2O as the product. This is why you breathe in oxygen. The reason you exhale carbon dioxide was because you broke it down from, you exhale carbon dioxide there and here, because you broke it down from glucose. All right, any questions about aerobic respiration? Because I got to get moving. All right, the last part that we talked about is aerobic respiration. Okay, we did this. We did this. You need to know what the respiration products are generally. I would know that aerobic respiration makes between 36 and 38 ATP. Most of that comes from the electron transport chain. You only made 2 from glycolysis. You made 1 ATP from each pyruvate in Krebs. So it's 2 from a glucose molecule. Most of that 36 to 38 is coming from the electron transport chain, right? We did amphibolism. All right, if we're going through this whole process in glycolysis, let me switch a color. I don't know what color. Let's say this weird pink color. If you are fermenting, you are only doing this. Just doing glycolysis, you're fermenting. So what you do is you made some electrons here. You made a little bit of ATP. That's your goal in fermenting. If you're fermenting, your goal in glycolysis is just the ATP. You don't care about the pyruvate. You're using the ATP. So you don't need NADH and its electrons. In fact, you need to get rid of it. So you feed the H and the electrons back into pyruvate. And that changes pyruvate into, depending on how you do it, you have a number of products you can make. You can make lactic acid, or you can make ethanol, or you can make carbon dioxide and or. This is where brewing makes ethanol, which is drinking alcohol, and sometimes makes bubbles in the form of beer. Beer is carbonated because the bacteria made the carbonation. You can also infuse it with more carbonation if you want, but that carbonation is natural. If you ever had kombucha and you've had a little bit of that fizz in there, that's natural fizz. You get that acid flavor, that's lactic acid. And it even has a little bit of alcohol in there too. Just a little bit. There is a little bit in there. So that's the idea of what fermentation is. What else am I supposed to talk about? Photosynthesis? Okay, briefly. Briefly. So I want to move on to the questions, and like I have lab at 6:45. But again, we took time on chapter 6, so I got to take time on chapter 10 for sure. Okay, so photosynthesis. Seems appropriate to use green for this one, right? What happens in photosynthesis is you have a membrane and you have some pigment molecules on the membrane, which are called chlorophyll. And this is all happening either in a chloroplast, if you are a eukaryote, or it happens on the inner membrane, also known as the cytoplasmic membrane, in bacteria. If you are a bacteria that can go through photosynthesis, like cyanobacteria, okay? You have chlorophyll, and then next to it we have, this should look familiar, ATP synthase. So what happens is sunlight activates the chlorophyll, that excites the pigment molecules, and when they get excited, it causes H pluses to move outside, and then we have pluses build up. They want to come back in, and then this allows us to make ATP. Should look familiar because it looks a lot like the electron transport chain, right? It works the same way, but instead of feeding electrons into it from NAD, we use sunlight to excite the whole process. It doesn't make very much ATP. It really doesn't. And so what happens is after that, plants use ATP that they make here in a lot of steps through a whole process. We'll take ATP plus carbon dioxide in a lot of steps to make 6-carbon glucose. This process here, we call them the light independent. We don't call them the dark cycle. It's the light independent cycle, also known as the Calvin cycle. So we made ATP in the light. And then without the light, we can turn that ATP and carbon dioxide into organic glucose. What do plants do with glucose? They can use it to build things for the cell. Of course they can, but what else can they do with glucose? Did you see what I did there? They can put it through glycolysis, the intermediate, Krebs, and the electron transport chain in aerobic respiration to make more ATP. Plants don't just have chloroplasts. All eukaryotes, including those that have chloroplasts, all eukaryotes have mitochondria. The only function that mitochondria have is the electron transport chain. That is it. Aerobic respiration. So plants do aerobic respiration too, to make more energy. They just happen to make more oxygen. Where do they make oxygen? In here, when they're making their glucose. Okay. The byproduct of photosynthesis. They just make more oxygen making glucose than they will use when they're breaking down glucose. Pretty inefficient, honestly. but it works for them. They seem happy enough. All right, so we're going to move on. Chapter 9's most missed topics. All right, the first one is osmosis and the tonicities. Osmosis is just movement of water across the membrane. And again, these slides have more information on them if you want to read them in detail. These will help you study on these topics more. But to remember, isotonic, we have the same inside the cell as we have outside. In a hypotonic solution, hypo means low, which means less. Less outside. Hyper means more outside. Typically. Okay. In a hypotonic solution, your cell has stuff, and the stuff outside the cell has no stuff, or has less. So the water moves in because the stuff can't move. And so the cell swells and can burst in hypotonic. In hypertonic, the cell is packed in stuff. So the water moves out of the cell shrinks. And this is a process called plasmolysis. All right, the question. Which of the following best describes the movement of water in an isotonic solution? In isotonic, we have the same inside and out, right? So does water move into the cell, causing it to swell? No. Does water move out of the cell, causing it to shrink? No. That would happen in hypertonic. The above happened in the hypo. Does water move in and out at equal rates, so there is no net change in volume? Or does water not move across the membrane at all? This requires understanding of chemistry. Most correct answer here is C. The water doesn't not move. It'll move in a little bit and move out a little bit always, because water is not stationary. The molecules are not stationary. So you have to understand about chemistry to answer this question correctly. C is our answer here. Next question. A patient is receiving intravenous fluids and the physician orders a saline solution to be administered. It's 0.9% sodium chloride. Which type of tonicity is this solution, and why is it chosen for the patient? This was the question in our chapter 9 activity. It's question 3, right? Or question 2. And question 3 asked us about pure water, right? So question 2 asked us, what is sodium chloride, 0.9% sodium chloride saline solution? We would want it to be isotonic, but is it isotonic because it maintains the balance of fluids inside and outside of the cell? Or is it isotonic because it'll allow the cells to shrink? It's going to be C, isotonic, because it maintains the balance of the fluids inside and outside. Even if your patient is dehydrated, we give them solution that would be isotonic to an otherwise healthy person, so we can bring their stuff back into balance, because your kidneys are meant to deal with isotonic situation. Isotonic for an otherwise healthy person. All righty. Now we're in chapter 25. Chapter 25, most missed topic. We have habitat range and transmission of diseases as a result of climate change. As the climate warms up, mosquitoes are moving north, right? The further north it's getting warmer, mosquitoes are moving up further north, and they're bringing with them the diseases that they transmit. You want to read more about how that works and how that affects public health. This slide goes into a little bit more detail on it. We're going to move on to the questions surrounding this information. How does global warming contribute to increased risk of mosquito-borne diseases. As the weather gets warmer, the mosquitoes move towards those now new warmer climates. They're taking up new environments. So does mosquito-borne disease decrease because breeding sites of mosquitoes will be lower because of rainfall? It's not A, that doesn't make sense. Does it reduce mosquito populations by lowering temperatures? No. Global warming raises the temperatures. Expands mosquito habitats? Yes, we're getting more of them as temperatures increase. And does it accelerate the development of mosquitoes in the pathogens that they carry? This tracks. Does it prevent the replication of pathogens within mosquitoes? If it really got hot enough, it could potentially work that way, but most of those pathogens are equipped for pretty high temperatures. C is our most correct answer here. Which of the following public health strategies is most likely to be affected by the expansion of mosquito habitats due to global warming? Well, we have reducing antibiotic prescriptions to prevent antibiotic resistance. This isn't relating to the global warming and how it affects mosquito habitats, is it? It might be an issue we might have to deal with, but not relating to mosquito habitats and global warming. Enhancing vaccination programs for mosquito-borne diseases in newly affected regions? Maybe. We have vaccines for some mosquito-borne diseases, and we're not vaccinating our populations against them, but we might need to if mosquito populations keep carrying their diseases further north. Promoting low-fat diets to combat obesity-related diseases? Not a bad idea, but not related to mosquito habitats or global warming. Increasing the use of personal protective equipment in non-endemic areas. Endemic means that the disease is natural in the area. Now, personal protective equipment. Might you need to increase personal protective equipment if we have the increases of disease? Yes, but the disease increases have to happen first. So that's why B is our most correct answer here. D is like getting too far ahead into the thinking and is very specific. It's not thinking about all of the big picture of it. Okay? Next, we're going to be getting into the chapter 10 questions. I think I have two. I have three for chapter 10, which I think we need the most anyways. First is the electron transport chain. To remind you, in bacteria, the electron transport chain is found on the plasma membrane, also known as the inner or cytoplasmic membrane. These are all the same thing. In bacteria, you have an inner membrane, and then maybe you have a big old thick wall around you, right? If you are gram-positive. If you are gram-negative, you have a inner membrane, a little thin wall, draw the wall all in one color, and then an outer membrane, kind of 3 layers. But the inner membrane, regardless of what type you are, the inner membrane here, that's where you're going to find the electron transport chain in bacteria. In eukaryotic cells, you have your cell, you have a nucleus where your little DNAs are, and outside of that nucleus, you've got these structures called mitochondria, and the mitochondria have an inner membrane. That inner membrane of the mitochondria is called the cristae of the mitochondria. They're the same, right? Remember, mitochondria likely evolved from bacteria, right? So anyways, that's the difference between those two. All right. All right. Question. Where does the electron transport chain operate in bacterial cells? Well, if we look back at our other picture from the previous slide, it happens on the inner membrane of the bacteria, which is also known as the cytoplasmic or plasma membrane. The inner membrane of the mitochondria is known as the cristae. That's where it happens in eukaryotes. It doesn't happen on the outer membrane of the mitochondria in anybody, or the nuclear membrane in anybody. It happens on the cytoplasmic or plasma membrane. In fact, bacteria don't have mitochondria and they don't have a nucleus. So it couldn't have been A, B, or D by default. It had to be C. Okay. You could have used process of elimination there. In the treatment of mitochondrial disorders, such as Leber's hereditary optic neuropathy, Which part of the cell is directly targeted by therapies aimed at improving cellular respiration? Obviously, this is affecting the mitochondria, and cellular respiration is another word for aerobic respiration, which happens in the mitochondria. And where in the mitochondria does it happen? The inner membrane of the mitochondria. We're talking about a mitochondrial disease. We're talking about a eukaryote, and eukaryotes have their electron transport chain for aerobic respiration on their inner mitochondria membrane. Next topic, talking about the generation of the proton motive force, keeping those H pluses outside the cell. The whole process of generating ATP is using the movement of electrons to force H's out and then let them come back in. These are chemicals moving. Instead of moving water across the membrane, we're moving chemicals across the membrane. H's, but they're protons, but still they're not water, right? So this is chemiosmosis, the movement of chemicals across the membrane to allow for generation of ATP through oxidative phosphorylation. Oxidative because it's electrons moving, and phosphorylation because we're phosphorylating, adding a phosphate to ADP to turn it into ATP. This is a little slide that breaks some aspects of that down. What is the primary role of the proton motive force in cells? The whole idea is you have a cell. Well, let me be more specific here. You have a membrane that has all those cytochromes and the ATP synthase. And the whole point is to build up pluses on the outside. And these pluses, just again, to remind you guys, this creates the proton. This is the proton motive force. The fact that we have pluses here means that they'll want to come inside. So it creates a gradient of protons. They keep wanting to come inside, and that drives ATP synthase to make ATP. So what's the primary role of the proton motive force? Is it to synthesize proteins from amino acids? You need ATP to do that, but you need ATP for a lot of things, not just that. Is it to drive the synthesis of ATP by ATP synthase? That's exactly what the proton motive force does. To facilitate the transport of electrons through the electron transport chain, the movement of the electrons through the electron transport chain drives making the proton motive force. So it's the opposite. To assist in replication of DNA, you need ATP to be made in order to make DNA. But that's not the whole story, right? So B was our answer. In the treatment of mitochondrial diseases, again, we're talking about making ATP, such as mitochondrial myopathy, which cellular process is directly impacted by therapies aimed at enhancing the proton motive force? So making sure that we have the pluses built up outside of the membrane. Why do we need pluses built up outside the membrane? So we can make ATP. Do you need ATP for this? Yes. You need ATP for this? Yes. Lipid metabolism. Are we breaking the lipids apart or are we making new ones? That depends. You might need ATP or you might generate ATP, depending on what we're doing with that. But our best answer here, we need the proton motive force, specifically so we can make the ATP. C is our answer. All right, and then we have the glycolysis, the intermediate step, which is pyruvate oxidation, and then the Krebs cycle and the electron transport chain. All of this is aerobic respiration. Go away so I can see what time it is. I've got 15 minutes, okay? All right, so all of this is aerobic respiration. This is a breakdown of the products and general descriptions of each of those steps of aerobic respiration. If you want to take the time to look over them, I'm not going to walk you through it. You guys can walk through it yourselves. We're going to move on to our question about this content. So again, glycolysis. In glycolysis, we want to make pyruvate. Then we have our intermediate step. where we want to turn pyruvate into acetyl-CoA. Then we have the Krebs cycle, where we're going to turn acetyl-CoA into NADH and FADH2, but not directly, by stealing its electrons. That's what these are. And then using those electrons in the electron transport chain. In your book, it's called the electron transport system. On the test, I'll call it the electron transport chain. The electron transport chain, we are creating that proton motive force by moving the electrons through the chain that lets us make ATP. Glycolysis will make us 2 ATP. Krebs will make us 1 for each pyruvate. We get 2 pyruvates from glycolysis, so a net of 2. And the electron transport chain makes 34. How many ATP molecules are produced directly during glycolysis from 1 molecule of glucose? Will you notice that 2 isn't one of your choices? That's because in glycolysis, you make a net of 2. What's really going on here is that you make 4, and then you use 2, which leaves you with 2. So B is our answer because of that. In patients with a condition, this is just for glycolysis, not for all of it, just for glycolysis. In patients with a condition that affects the mitochondria, where your electron transport chain is, such as mitochondrial myopathy, which stage of aerobic respiration is most directly impacted in terms of ATP production? Again, the mitochondria, that electron transport chain is happening on the inner membrane of the mitochondria called the cristae. All right. The electron transport chain is your glycolysis and your Krebs. I don't know why I'm not thinking why there. And Krebs. Glycolysis and Krebs happen in the cytoplasm. Again, period. Whether you are a bacteria or a eukaryote, glycolysis and Krebs happen in the cytoplasm. So does your intermediate step. Glycolysis, cytoplasm. Pyruvate oxidation, which is your intermediate step, cytoplasm. Citric acid cycle, also known as Krebs, cytoplasm. Electron transport chain, mitochondria. What happens? What's dealing with mitochondrial stuff and ATP production? The electron transport chain happens in the mitochondria. All right, do you guys have any questions for me? I have 10 minutes to answer some questions before I have to head to my lab. Do you guys have any questions for me? All right. I don't know. I'm assuming you guys aren't asking questions or I would hear it, right? So I'm not hearing anything. We don't have this muted and you guys have been asking me questions about it.
Questions, thanks.
Oh yeah, okay, I heard you. Okay, cool. Okay. All right. I wasn't sure because it's been quiet the whole time. And Gracie's been talking in the chat, but yes, okay. So if you guys don't have any questions, then that is great. And that's going to be it for our review today. Again, I'll post this in your discussion areas. And otherwise, thank you guys for showing up. And happy studying. And I will send out a reminder to attend either virtually or in person on Monday or Tuesday, and the dates and times and everything like that for that, and a reminder with a link and all of that. So if you guys have any questions while you're studying, email me. And don't forget to use the STEM Lab or the SSOC as a great study resource to prepare you guys for your exams coming up. All right, but that's it. Thank you guys for joining, and I'll see you. Thanks, guys.
Bye.